Now that we've talked about some basics of viruses, let's work through the process of how viruses replicate themselves within their host cell. We're going to go through the basic process of the viral life cycle, how a virus infects its host and uses the host cell's organelles to replicate itself. The first step is attachment. The virus binds to molecules on the surface of the host cell. The right molecules need to be present for the virus to bind, so not all viruses can infect all types of cells. Next, the virus enters the host cell. Different viruses do this in different ways. We'll talk about that a little more later on. As the virion enters the host cell, the capsid comes apart, spilling the virus's genetic material into the host cell's cytoplasm. Next, the virus uses the mechanics of the host cell to replicate its genome and produce viral proteins. Now that new viral DNA or RNA has been produced, they come together and are enclosed in a new capsid to form new virions. Finally, the newly formed virions leave the host cell and are now able to infect new cells. You may want to go back through that process a few times and make sure you understand it. When you're done, try to answer this question. Remember that viruses aren't considered cells and don't have their own plasma membrane. However, there are some viruses that are surrounded by a phospholipid bilayer. But first, some terminology. Viruses that aren't enclosed in a membrane are known as naked or unenveloped viruses. Enveloped viruses are enclosed in a plasma membrane made of phospholipids. However, they don't make that membrane themselves. Instead, they steal it from their host cell. Think back to the last step in the viral life cycle. Once the new virions assemble, they need to leave the host cell. Some viruses do this in a process called budding. The new virus, or virion, assembles near the plasma membrane of the host cell. The membrane starts to bulge out. This is called budding. Instead of the virus breaking through the membrane, the membrane around the virus pinches off and closes behind it. This leaves the virus enclosed in a bubble of the host cell's membrane. Now, when that enveloped virus wants to enter a new host cell, it can do that process in reverse. The viral envelope can fuse with the host cell's membrane and let the virus sneak into the cell. Viruses that don't have an envelope can't use budding and membrane fusion to get into and out of host cells. Instead, many of them punch through the host cell's plasma membrane and inject their genetic material. The problem with this is it can sometimes cause cell lysis, or the splitting open of the host cell, which causes the cell to die. And remember, the virus wants the cell to stay alive at least long enough for it to replicate itself. Just like how there are pros and cons to being a DNA virus or an RNA virus, there are pros and cons to being an enveloped or unenveloped virus. Enveloped viruses have several advantages. They can enter and exit cells without causing cell lysis. Also, because they are surrounded by plasma membrane from a host cell, they can go incognito. It's harder for host cells to recognize them as a virus, so they can better avoid the host's immune system. Finally, the envelope gives an extra layer of protection around the viral genome. But unenveloped viruses typically can only infect cells that don't have a cell wall. In cells like plant cells, the cell wall is a hard covering outside the plasma membrane. So the cell wall prevents enveloped viruses from fusing their viral envelope with the cell's plasma membrane. Unenveloped viruses can enter and exit by punching through the cell wall, so they have an advantage in infecting plant cells and other cells with a cell wall. So scientists think that the viral envelope evolved as an adaptation for infecting animal cells, which don't have cell walls. So one of the differences between unenveloped and enveloped viruses is that unenveloped viruses are more likely to quickly kill the host cell by cell lysis. Because they kill more host cells and kill them more quickly, they also tend to be more virulent or cause more severe symptoms in the infected organism. Unenveloped viruses are also more resistant to extreme conditions and things like disinfectants, because their outermost coating is the capsid and not a sensitive plasma membrane. Think back to the first step of the viral life cycle. We mentioned that viruses need to bind to specific molecules on the surface of the host cell. So not every virus can infect every type of cell. Viruses are specific, not just to the species or host cell, but also usually the tissue type. So a virus can infect your lungs, but may not affect your digestive system. Again, that specificity is because the virus needs to recognize the right cell by binding to specific molecules in the cell's plasma membrane. For many viruses, the molecules that bind to molecules on the host cell are glycoproteins. By now, we're probably all pretty familiar with what the coronaviruses look like. You may have noticed that they are covered in little spikes. These spikes are actually the glycoproteins of the coronavirus. Now try to answer this question to test your understanding of why viruses are specific in the types of cells they can infect. While viruses are specific, you're probably aware that viruses can jump between species. To understand how this can happen, let's take a look at the example of bird influenza and how it jumped to humans. Influenzas, or flu viruses, that infect birds have glycoproteins that don't bind well to the receptors on human cells. So those viruses cannot infect human cells. However, those glycoproteins do bind to the receptors on pig cells. Human influenza viruses also bind well to pig cells. So a pig cell can be infected with either a bird influenza virus, a human influenza virus, or both at the same time. Influenza is an RNA virus. Remember that we talked about how RNA replication makes more mistakes? If a pig cell is infected with both influenza viruses at the same time, some mistakes can happen in RNA replication that create a new strain of the bird influenza that also has glycoproteins from the human influenza virus. This new strain of the bird influenza can now infect humans because it has glycoproteins that can bind to human cells.